And so for those of you that don't know me, I'm Kate Laskowski. I'm a faculty in the Department of Evolution and Ecology. And today I'm delighted to be able to introduce our STORR speaker, Dr. Ben Sheldon. And so Ben Sheldon is an evolutionary and behavioral ecologist who is broadly interested in understanding the causes and consequences of individual behavioral variation, a topic near and dear to my own heart. And so Ben got his bachelor's from Cambridge, his PhD from the University of Sheffield, and then did postdocs both in Uppsala and Edinburgh. He then started his position in Oxford in 2000, where he's been ever since. And Ben's career is one that I can really only hope to aspire to. He's continually received funding to support his work and his group. He's earned many awards and prestigious appointments, including being the first holder of the Luc Hoffmann Chair in Field Ornithology, the director of the Edward Gray Institute, the Zoological Society of London Scientific Medal, and just so you know, he's not completely UK biased, he also got the American Society of Naturalists Distinguished Naturalist Award. He also has a Wikipedia page, which I think speaks to his international stature. And so Ben's work is focused on taking advantage of an incredible study system that we're going to hear about today, the great and blue tits of the Wytham Woods in the UK. And so this population of small birds has been continually studied since 1947, which is pretty amazing. The system now has over 1,000 nesting boxes where all animals are tagged and followed throughout their lives, providing information now on over 40 generations of birds. Using the system, Ben and his group have been able to provide incredible insight into the long-term patterns and changes in these populations, including shifts in breeding date due to climate change. Importantly, as with all of Ben's work, he goes beyond just describing patterns to instead dig into the details and figure out the mechanisms. And with the tits, this earlier breeding seems to be tied to the emergence of their preferred food, caterpillars. And so Ben's work has not just looked at changes in one species, but also digs into how entire communities are being reshaped by our changing planet. And then finally, get my notes here. On a personal note, Ben has also proven himself to be a deeply supportive and kind person. As some of you may or may not know, when I started here at Davis, I was faced with the realization that a former collaborator of mine fabricated a bunch of data that he then sent to me to write up as part of a collaborative project. Once I found out, I publicly retracted these papers and inadvertently stirred up the biggest fraud scandal in my field in memory. Needless to say, this was a terribly dark time in my career. And Ben, who I had never met or really interacted with, was kind enough to reach out to me and basically just say, hey, stay strong, you're doing the right thing, and let me know if you ever need a letter, because I'll be happy to write you one. And this really mattered to me. I mean, I was a first-year faculty, wondering if I'd keep my job or what my career would even look like from this point forward. And here was this massive figure in the field, basically telling me it was going to be all right, even though he had never met me. And I took him up on his offer. I don't know if that was good or bad, or if he appreciated that or not, but I did. And he has now written me letters for several grants and awards, and most recently, I'm told, for tenure. And they've all been successful. And so this is all from someone I had never even met. And so I'm delighted now that the STORR Endowment has allowed me to bring Ben here to Davis so we could finally meet in person for the first time, and I could thank him for his kindness. He's had an immensely positive impact on my career, and I am deeply grateful. And so with that, I very much look forward to hearing more about his birds, and so please join me in welcoming Ben.
Thank you. Thanks, Kate. Thanks very much. Can you hear me okay at the back? Yeah, great. Thank you. So just about the Wikipedia page, so no editing it while I'm talking, please. So, okay. Anyway, thanks very much for the invitation. It's really been a great honour to come here and talk to you. I've had a fantastic time so far. Got lots more meetings tomorrow as well, so really looking forward to that. What I'm going to try and talk to you today is about a particular type of study, which is long-term studies of individually marked animals in natural populations, and to try and convince you that they're a very powerful technique to try and understand a whole range of questions in ecology, evolution, and behaviour. And I'll focus mostly on the work that I've been involved in myself, but also talk a little bit about some of the other work done in similar kinds of systems using the same kind of approach as well. Now, obviously, the idea of kind of long-term studies in ecology and evolution isn't a new one. We've got some amazing long-term time series, this incredible time series of Kyoto cherry blossom timing for over 1,000 years, which you can link to kind of climate change and so on. Or we've got this awesome study that was set up by Richard Lenski, a sort of former Storer lecturer, which has run for over 75,000 generations of E. coli under standard conditions. So this has been, particularly with the advent of genomic sequencing to look at changes in these populations, has taught us an immense amount about evolution under standard conditions. But I'm going to talk about studies that have a sort of crucial difference from those two types of studies, and that's studies that rely on a very simple kind of bit of technology, which is knowing about the identities of individuals, right? And this turns out to be something that unlocks all kinds of scientific questions and enables us to answer those. So it's really kind of fundamental to the study I do and lots of other studies like it, that we know who individuals are and can then track them and their links to other individuals through time and through space. So what does individual identity enable in long-term studies like this? So there's a bunch of things here. So it enables you to quantify age and age structure because you can recognize individuals. You can ask about the linkage between life history stages. You can measure biological fitness over lifetimes. You can use that to work out measures of sexual selection or natural selection and replicate those over time. You can dissect the causes of phenotypic change. You can link between generations. And if you know who individuals are and who they're interacting with, you can quantify social structure. I'm going to try and touch on examples of all of those, some of them combined together over the course of this talk. So I'm going to try and pack in quite a bit into this talk. So that's the introduction done already. So I'm going to start by talking a little bit about history. You can't really talk about these kinds of long-term studies without acknowledging some major historical context. We're going to talk about three particular applications of those studies. One to look at sort of understanding climate change, natural populations. One is their use for understanding inheritance, selection, and particularly the impact of rare events in natural systems. And then lastly, some work that we've been involved in looking at sort of behavioral and cultural evolution in populations that's relied on that individual identity data. I'm going to talk a little bit about the future prospects and challenges for these kinds of studies as well. So history first. So the pioneer in this area was a woman, Margaret Morse-Nice, who may not be very well known sort of outside this particular sort of niche area that I work in, but she was really the first person to do work on individually identifiable wild birds. So she's studied for about 8 years a population of song sparrows in Ohio. And she published a massive amount of data in this very, very long paper, a 300-page paper in the Transactions of the Linnean Society. This has some amazingly pioneering kind of work in it. So for example, the figure here is an illustration of little pedigrees that she'd built up from her study, mapping the inheritance of migratory behavior, so whether birds were migratory or non-migratory, and basically showing based on these pedigrees that actually this couldn't be something that was inherited as a simple single locus trait, even though it was a sort of a bimodal behavior in the population. So really kind of pioneering kind of work. Second pioneer in this area, sort of much more local to me, a man called David Lack, who was appointed as the director of the Edward Gray Institute, which I'm currently director of, in 1945. And he was already a sort of an established kind of name in the field. So he'd carried out this famous study where he'd color-banded individual European robins and characterized their territoriality and individual behavior and so on, before he was even appointed to this position. And he was also, as we'll actually see a bit later, he was a crucial person in bringing Darwin's finches to kind of the prominence that they currently have today. And so Lack moved to Oxford, and he was looking for a kind of study system to kind of collect some basic data about population biology, things like how long individuals live, how many offspring they have, all these kinds of things, which just weren't known in those days very well. And in 1946, he visited some colleagues in the Netherlands, visited a guy called Hans Kluiver. This was actually an attempt to persuade Nico Tinbergen, who was based in the Netherlands, to come over to Oxford, which he subsequently did. And of course, Nico then won the Nobel Prize for his work in ethology. But while he was there, he met these people in the Netherlands who told him about the work they'd done studying great tits as this kind of model species. And that's really where Lack got the idea from. And so he established his population in 1947. And then after a decade, bequeathed that population to Chris Perrins, who carried out his DPhil work at Wytham in the late 50s, early 60s, and actually expanded the study to kind of basically the scale it is today. So just over 1,000 of these artificial nest boxes, which the great tits prefer to breed in over natural cavities. And so this is a picture of David Lack and Chris Perrins together at Ithaca in the States, inspecting a chimney swift nest from the early 60s. Okay. No? Oh, okay. And one of the sort of nicest things for me about being involved in this study myself is that Chris Perrins is very much still alive and still very interested. So I think he's been basically thinking about the biology of tits for 65 years. So he's one of these people who's got this amazing fount of knowledge. And so to be connected by basically sort of one step to a person like David Lack is also kind of a tremendous privilege for working in this study as well. So, okay. I keep going, right, great. So it became pretty apparent early on that there was this kind of value in the long-term nature of these kinds of studies. So this is the first sort of major write-up of this work by Lack in 1964. And he wrote at the time, as the Marley study, which is where they started the Wytham study, is only in its 17th year, there are many points on which it's not yet possible to draw firm conclusions. And then he goes on to say that the 15th year was, for example, very different than the rest and so on. So this idea that with long time series, you could see events that were unusual and you needed to see these to make sense of what was happening in populations was a thing that quite quickly became apparent. And I'm not really sure quite why it is, but around the same time, there was a whole set of other people who were founding population studies that went on to become long-term studies. So some of them are on birds, like these studies here. So these include Florida scrub jays and acorn woodpeckers in California and song sparrows off the coast of British Columbia. And then also around the same time, some long-term mammal studies started. So primarily studies of primates, ungulates, some carnivores, and so on. So a whole bunch of studies started in the sort of period between about 1960 and 1975. And it's really this set of studies that's kind of the inspiration for this talk today, which I'm going to illustrate, particularly with the Wytham work. Okay. So I'm going to introduce you a little bit to Wytham Woods, this study site. So this is a site in southern central England, so just west of the city of Oxford. So the Wytham estate was given to the university in the 1930s, and it really became a place where a lot of sort of early ecological work was done. So, for example, Charles Elton carried out a lot of basic work on population dynamics of rodents in Wytham Woods, and was a sort of natural place for people to start this kind of study. So it's about 400 hectares on a hill just west of the city of Oxford. So it's a beautiful place as well. If you're ever in Oxford, get in touch with me and I'll take you up there. I love nothing more than an excuse to go up and show people around the woods. But since 1947, and really in its current form since about 1960, we've had over 1,000 nest boxes in fixed positions throughout the woodland. And in some ways, the data collection that underpins most of what we do, the core data is very simple. It's just based around going around inspecting nest boxes on a regular schedule and collecting data on which ones are occupied, by which species, how many eggs they've laid, when those eggs hatch, how many young survive, and so on. We also catch and mark the parents and the offspring. So it's kind of basic standard field data collection, often supplemented with other approaches and other techniques. But over the years, that kind of basic data, baseline data, builds up to a kind of large data set where we can say lots about variability in the population from different sources. So we had our sort of 75th anniversary a few years ago. So one of my students made some really nice infographics to summarize what we've seen in this population. So various sort of facts and figures there. I think one of the things that's kind of remarkable for me is that by ringing, or banding, as you say over here, of course, by ringing the parents and the offspring together, we can track individuals back over as many as 35 generations. So you can catch a great tit today and in principle might be able to trace it back to an individual that was alive in 1960, which is obviously well before even I was born. And so it's kind of a sort of sobering thought that genealogy, that lineage is still persisting in the population. On all of this data and with supplements to it, a massive amount of work has been done. So again, we sort of totted up various things. So you can see 367 papers, 71 PhDs and so on. A lot of field work, so about 500,000 kind of checks of nest boxes, about 100,000 kilometers walked and so on. I can't do justice to all of that work in this talk, you will understand. But but I think it's probably true that many of the other long-term studies that I've shown have similar bodies of work that's the product of similarly intensive, dedicated field work. So it's kind of non-glamorous work, but it generates massive amounts of useful data. Okay. So I now want to talk a little bit about some of the things we do with this study and the kind of applications of it. So I've really picked sort of 3 things that we can talk about that these studies have been kind of influential and informative in helping us to understand. First is understanding responses to climate change. The second is telling us about mechanisms of inheritance or the strength of selection in natural populations, and particularly how rare events can be important in understanding that. And lastly, work on sort of behavioral and cultural evolution, processes that arise due to interactions between individuals and the transmission of information between individuals. So it's sort of hard to believe it was as recently as this somehow, because it's an issue that's very much to the fore, but it was only in around 1990 that the IPCC, the first assessment report, was published and really sort of started to flag this issue that there could be major issues for natural terrestrial ecosystems due to climate change. And what they said at the time is obviously the rate of projected climate change is the major factor determining the type and degree. These rates are likely to be faster than the ability of some species to respond, and responses may be sudden or gradual. And I think at the time we knew very little about actually how these things would play out, and it's long-term studies sort of like this, the one on the Great Tits, that has told us a bit about the different ways in which populations can respond to these kinds of changes. And you could plot a figure like this for dozens, if not hundreds of studies now. So you can plot some standardized event. In this case, this is the average egg-laying date of the population in each year, plotted over time. And you can see that there's a very clear decline in that data. So the birds are breeding earlier now than they were at the start of the study, and earlier by about 2 weeks or so. This is a typical pattern in this field, though. There's quite a lot of sort of variability from year to year, but the overall downward trend is very clear. So obviously that's data sort of up to the most recent data that we've got the average for. At the moment they're laying eggs, so we don't yet have the average for 2025. Now, how early was that signal detectable? We might want to ask that question. So with the benefit of hindsight, could we have seen these signals earlier than the IPCC was flagging the potential for these? And so what I plotted here is basically the slope of the regression of that egg-laying date on time over year when you sort of truncate the data sets at different points. What you can see is actually that before about 1990, actually, there was very little signal in the data. After that, the signal has basically converged on a fairly stable kind of estimate of the rate of change over time. So the birds are breeding about 1/4 of a day earlier per year. And that's the sort of summary over those 6 decades. One of the things that I'm not going to show you, but is also that this turns out, particularly the early part of this signal, what you see depends a lot on when you happen to start measuring the system. So actually, it turns out that about the time that this concerns about climate change were being raised was about the time we were first able to detect these things in natural populations. So it's not a signal that we could have seen much earlier than that. Now, if you see a trend like this over time, a change over time, so one of the questions that really one might ask is what causes that kind of shift in a population? And basically there are sort of 3 mechanisms that can produce a population level change. It could be any single one of these or any combination of these. So it might be that what we're seeing is adaptive evolution, so in other words, differential survival of genetically different individuals that's producing a population change. It might be that this is what we call phenotypic plasticity, so the individuals producing different phenotypes in different environments. Or it might be due to migration, so individuals with different phenotypes redistributing themselves around in the environment. And actually working out which of these 3 mechanisms and how important they are for producing long-term change is kind of impossible unless you know about individual identities. So these sorts of studies have been very useful for helping us understand those kinds of questions. And at about the same time as people were getting very interested in climate change effects in populations, there was a great deal of interest also in the development of an application of techniques using quantitative genetics in natural populations. So these kinds of data where we're trying to estimate genetic and environmental components of traits in populations. And so that kind of coincidence meant that there was lots of people with new tools, wanting to apply them to these kinds of data to understand why these changes were happening. So a great slew of papers published talking about different types of response in different kinds of populations. I'm just going to go into a little bit more detail, sort of our understanding of what's driving this change in the great tit population that we've seen in Wytham. And like most systems, when you're looking at change over time in response to climate, this actually involves also understanding linkage between different trophic levels. And the sort of simple system that we think about often is, you can think about it as just involving deciduous trees, exemplified by the oak tree, insects that eat the leaves of those trees, exemplified by the winter moth, and then birds that eat those insects, exemplified by the great tit. So we've got this nice tritrophic system, and we can actually address quite a lot of sort of what's happening at different levels in this system, in this population. So what happens here is that the trees come into the leaf, bud burst state, sometime in the spring. Caterpillars hatch out of their eggs to feed on those newly emerged leaves, and provided that they hatch at the right time, they will develop and grow into large fat caterpillars. And the problem for the birds is that they have to lay their eggs, in the sense, to anticipate the point at which there'll be this abundance of large caterpillars in the environment. And it's about 30 days from when the great tit, lays its first egg to the time when its chicks have their peak food demand. So there's kind of problem in terms of kind of synchronizing your behavior to an event in the future. And of course, the problem is that from year to year, bud burst state varies. So this is a moving target that varies every year. And of course, the caterpillar dates will vary as well. Now, one of the things we know is that we've seen this kind of obvious increase in the early spring temperature in this population over time. And that's as the average egg-laying date of the great tit population has advanced. Almost whatever type of simple climatic index you kind of characterize this population with turns out to be a very good predictor of the population average laying date. So this is a measure called warmth sum. Doesn't really matter what that is, but you can see there's quite a strong correlation between the population average breeding date and the warmth sum in each individual year. So if you're an ecologist, kind of studies from natural systems with R-squareds, 72% is kind of like, well, this is freakily strong. So a very clear thermal effect. And actually, the same climatic variable also predicts the timing of the winter moth abundance in the population as well. Again, each of the dots is from a different year in the population. And so because they're both responding to the same environmental variable, then what we see is if we plot the date of the great tit laying and the date of the caterpillar peak abundance in each year, we see a nice correlation between the two. So these two things remain synchronized over time. So this is a case where there's been a shift in the birds, but it's also tracked the shift in the timing of the peak abundance of their prey. And so, as I said, lots of these long-term studies have found similar shifts in phenotypes over time. So this is just for red deer, another one of these classic long-term studies showing lots of different traits that have advanced in their timing over the years. And these studies allow us to kind of look at those shifts and say, what are the mechanisms that underpin these shifts at the population level? So for great tits, the answer, it turns out, is very simple. So what we were able to do by having individuals studied in multiple years was to say, okay, we'll take this individual and compare its behavior in 2 years that differ in their temperature. We'll ask how much it shifted its behavior between those 2 years. And that's what's plotted in the left-hand figure, is the shift in dates within individuals against the differences in the temperature in the 2 years they're observed in. And we estimated the slope through those data, and you can see that the slope response within individuals is almost exactly the same as the slope of the response at the population level. So the population level response can be entirely explained by individual plasticity. So there's no need to invoke anything like adaptive evolution. This is just birds responding to temperature fluctuations. That's not the case with all of these long-term studies. So for example, this was a really nice study where these changes in timing in the red deer population were partitioned due to effects due to changing demography, that's changing age structure really, plasticity like in the great tits, but also estimated changes of the genetic component effectively of breeding time. And it's a bit of all three of those in the case of the red deer. So it seems to be both plasticity, demographic change shifting the population mean, but also genuine evolution of breeding time as well. So that's a sort of, I guess, many different types of answer to this question about what underpins climate change responses in different types of population. Okay. Second thing I want to talk to is really about how we can use these kinds of studies and linking generations to tell us lots of interesting things about what's happening in populations. And so here we've got a nestling great tit again, getting its individual ring that's going to enable us to identify it and link it to its parents and to its offspring if it has any, and so on. We can use those data and put them together to build a pedigree, a multi-generational pedigree that links together individuals. So this is just a plot of the pedigree of the Wytham population, sort of DPhil student of mine, Caris Jones, who's been building that. And there's a little bit zoomed in that shows you sort of individual relationships in this pedigree. So lots and lots of data about how individuals are linked together. And of course, they're distributed across lots of environments. So we can use those to try to tease apart the causes and consequences of the phenotypic variation we see in the population. One of the things that Caris has shown is that there's a very interesting effect of sort of shared environments on the similarity between parents and offspring. So the figure here on the left here shows a series of different models where what she's done is to ask how important the spatial similarity, the sort of being close in space, is to contributing to phenotypic similarity between relatives, or how important environmental similarity is. And you can see that these are relatively big chunks of the variance is accounted for by these processes. And a much easier way to visualize that is just to use a very simple comparison, which is to say, okay, if we've got mothers and daughters, sometimes the mother is, and where the daughter settles is very close to where the mother sort of laid the egg. Sometimes she disperses far away. So this is the dispersal distance in the population. We just split those into 3 equal groups and look at the heritability, the similarity between mother and daughter breeding time. You can see as you go from short to medium to long distances, the daughter's breeding time becomes less and less like the mother's. So there's this kind of, what we expect to see is as distance increases, environmental similarity declines, and that's probably what explains that decline there. So that we can use these techniques for asking about the causes of variation in populations, but they can also tell us things about other aspects of mating. So one of the sort of nice examples of this is using them to look at what happens when you get relatives that breed together. And this is a classic case of something which is actually quite rare, but potentially quite important for informing our understanding of things like how far individuals disperse and so on. So a PhD student of mine, Marta, a few years ago, worked on trying to look at the strength of inbreeding depression in the Great Tits and Wytham. And you can see here the summary of how many individuals were able to identify where we knew all the grandparents. We could work out something about how closely their parents were related. And you can see that, roughly speaking, about 1% of breeding events, roughly about 1 each year, were closely inbred. So if you were a PhD student sent out to study this problem for 3 years, you might, if you're lucky, find 3 or 4 cases. Obviously, pooling together over a much longer time series, we had a much larger data set to investigate. And one of the things that showed us is that measuring inbreeding depression and measuring it over the full life cycle and actually into other generations as well, that inbreeding depression was perhaps much stronger than we thought. So here what we're doing is we're looking at how fitness declines to do with as the individuals that produce offspring are more closely related to each other. And what the figure does is to compare the fitness of outbred birds, the grey bars, with the fitness of the closely inbred birds at progressive stages through life history. So this is the number of young produced, number of fledged, number recruited to the breeding population, the number of grand offspring that are produced by those individuals, and the number of grand offspring that are then recruited to the breeding population. So we've now gone 3 generations. And what you can see is that there's an effect of inbreeding seen even sort of several generations removed from the inbreeding event itself. So in other words, the effect compounds and is much, much stronger than we might have estimated had we just focused on the effect on numbers of young fledged from the nest or so on. So it's quite hard to talk about the application of genetics and selection to these kinds of studies without actually talking about perhaps one of the most famous studies of this kind, which is this famous study of Darwin's finches. And actually there's a sort of connection to the Great Tits study, which is not maybe as well known as I think it should be. So these are, of course, Darwin's finches, which I think probably everyone knows, the sort of the textbook example of evolution in action. Finches that were collected by Darwin in the 1830s in the Galapagos Islands, actually not that influential in his thinking, partly because he didn't really sort of understand quite where he was collecting individuals and so on. And actually, it was really David Lack who founded the Great Tit population study who coined the term Darwin's finches and published a book in 1947 which laid out lots of ideas about the way in which interactions between ecology and evolution could help us to understand the sort of distribution of species on different islands in the Darwin's finches. And the book was reviewed by Richards a year later. And Richards, I think, perhaps was not super impressed by Lack, he'd been to Galapagos for a few months and measured some specimens in the museums in California, and said the problem of whether certain specific characters are or are not adaptive is one involving studies of field populations, genetics and variation, not one, I think, to be settled in a 4-month stay, even if supplemented by the examination of skins in the museum. And it's, in a sense, we're fortunate as a field that two people, Rosemary and Peter Grant, actually took on that challenge of conducting a long-term population study of evolution of Darwin's finches on Daphne Major Island, which they did for about 35 years. So these are really kind of landmark studies. And one of the things that they illustrate is how data can remain relevant for a very long period after it's collected. So this was, for example, one of the first clear bits of evidence that you could predict evolutionary responses in natural populations was the severe drought in 1977, which caused a shift in the distribution of offspring, which was due to differential survival of the parents through that drought. Run that study for another 2 decades, and you have another drought. And so the first drought is in 1977. You see the beaks evolve to become larger. That's when there's a small competitor species present. Run it another 20 years, another drought, this time with a larger species present, and what you see is beak size evolving in the opposite direction. So in other words, great example of showing us how ecological interactions are important to tell us about the direction evolution is going to take. But what I thought was really impressive, and I wanted to mention, is that the Grants had the foresight to collect blood samples from almost the entire time series they were working on these birds, at a time when the idea of what you can now do with a blood sample in terms of genomic analysis was just kind of not even on anybody's kind of remotest sort of dream. And so there's, I think, a really impressive paper. I've got nothing to do with it. I just think this is a great bit of work. So if you haven't read it, I really encourage you to read it, by Eric Embody, which uses 30-year time series, sequences 5,000 or so genomes from the Darwin's finches, and shows that at a bunch of loci that seem to be important in affecting beak size, that there's been a shift in allele frequency that corresponds with the phenotypic change in that drought. So a beautiful example of kind of long-term evolutionary change with a potential genetic mechanism. Okay. So I want to switch now and talk about my third area, which is the study of kind of social structure and cultural evolution. And this is work that's been facilitated in our case by a second kind of mark that we put on the birds. So not just the metal ring, but this little plastic ring on the leg, which is a tag, PIT tag or an RFID tag, which is inert, but if you get the birds close enough to an antenna, you can detect those birds. And we started a program about 15 years ago of tagging lots and lots of birds and putting out a grid of feeding sites where the birds would come to. So we had sort of 65 sites and a grid across the whole of Wytham Woods. And this is what this looks like in real life. So you've got a logger, it's inside a cage to keep squirrels out, because otherwise they eat all the equipment. And you've got, each time a bird comes in, you'll see that a little code flashes up, and that's that bird's individual identity code. So put these out in the woods, they opened automatically and so on. You get lots and lots of data of who the individuals are. And yeah, so here's a great tit. Blue tit there. And that's marsh tit, kind of, yeah, very cheeky thing. So, you can imagine we were tagging several thousand birds and putting these out. You start to get a lot of data, sort of detections of birds, and you can build these up to identify flocks of individuals that flock together. We use those to build social networks, telling us about which individuals were being seen statistically more often together than we would have expected by chance. And from those patterns of association, we can build social networks for these birds. So former graduate student Damien Farine, who I know spent some time here at UC Davis as well, he was very involved in this work. So this is a social network he built. Actually, it's a multi-species social network showing how the different species are interconnected in a part of the population. But we were really interested in trying to use this kind of data and approach to understand something about the way that behavior could change in a population over time. And this was inspired by this classic example of tits opening milk bottles, which it's from nearly a century ago, but this was a documented spread of a novel behavior in these birds in the wild. That used to have milk delivered to doorsteps, and the tits learned that if they pecked through the foil, they could get cream out. This inspired an experiment by a doctoral student and then later postdoc of mine, Lucy Applin, to see if we could actually experimentally recreate that process and understand it in a natural population. And Lucy's experiment involved, in a sense, training wild-caught individuals to solve a puzzle, and there were two solutions to that puzzle, so action A or action B, and those had an equal reward, and there was a control group which were taken into captivity but not taught a solution to a puzzle. And once the birds had learnt the solution, they'd be released into the wild together with these puzzle boxes that would log what birds were doing on the individual puzzle boxes. So we set this up with 8 different sort of replicate subpopulations, where we seeded 3 with the sort of individuals knowing, call it the red solution, 2 with the blue solution, and 3 controls. And these are just some short videos of, I'm going to click on that one. Here we can see a bird that knows how to do this. This is the blue solution. Gets a mealworm. So it's sitting on an antenna that records who it is and so on. And now the device closes, so it's ready for another bird. So it's great. You can collect lots and lots of data this way. Just to show you, this is the other solution. This is the red solution at a different site. So a bird that knows how to solve it. This is really interesting. A bird that's learned that if it comes down quickly enough, it can scrounge the reward. Then here's a bird that's still thinking about, hasn't worked out the puzzle yet. So we put these out in the field, and we could track what happened in local populations over time in relation to what we'd seeded in that population. And so the first thing is to look at what we see in the control populations. So here we see the spread of the if we look at the proportion of the population that was able to solve the puzzle from 0 to 100%, eventually a bird spontaneously discovers that there's food in this box, starts opening it, and then it starts to spread in the population. So great tits are really good at kind of innovating naturally. So we see a natural diffusion there. It's really striking, though, if you compare that versus the experimental populations, how much faster it spreads through the experimental population. So it reaches up to about 80% of the population in just 20 days or so. And the real test, though, is what the individuals are doing in relation to what was seeded in their local population. So here we can see what happens in relation to the side bias that was introduced. We've got, remember, control either pushing on the blue side or pushing on the red side. You can see that there's a very close match in the experimental replicates between what was seeded and what actually spread through the population, whereas the controls actually, there's more or less a 50-50 mix because nothing has been seeded there. So this is really strong evidence for kind of social learning of this specific solution in the population. By taking the sort of social networks that we'd built, we could plot how that behavior had diffused through that population. So the yellow dot was where it was seeded, and as the dots go red, individuals have become knowledgeable. And this kind of model then allows us to actually estimate, for example, what the effect of having a knowledgeable neighbour is on how likely you are to learn the behaviour. And there's a massive increase in the likelihood of learning if you're next to a knowledgeable neighbour. Okay, so one question is, we've introduced these behaviours, do they persist in the long term? So what we did was to go back the following winter. So there'd been a short experimental period at the start of the year, then the breeding season where there's lots of mortality, lots of new birds born and so on. So we've got a population with lots and lots of naive birds in it. We just went out and put the devices in the same places where they'd been tested experimentally. And there's a really striking, clear persistence of this bias in each of the experimental sites and a very rapid rediffusion. In fact, it spreads even more quickly the second year than it does in the first year, perhaps because there's rather more knowledgeable individuals still in the population. So it's a really nice example of sort of cultural evolution in a population. I'm going to finish by one slightly more recent example of related work. And so this is actually using a behavior that sort of occurs naturally. And this is some work done by again, a graduate student, our current postdoc, Nilo Marino Caldo in my group. And Nilo was very interested in the question of how we could sort of think about sort of learned behaviors and how they vary at a small scale in populations as a function of demography in the population. We know individuals are moving around and they're potentially taking information with them. That varies in space, that's sort of those demographic processes. Could we use that variation to help us understand the small-scale variation in kind of song diversity in this population. Great tits have this characteristic sort of variable song which you can record lots of. So this was vast amount of work. Nilo recorded 23,000 hours of recordings. He did some fairly kind of fancy machine learning to process these data to kind of characterize individual songs and so on. And we're able to use those data to try and estimate how these demographic processes contributed to variability in song in the population. So basically, if we go down to the sort of neighborhood level for the birds, we can measure things like the amount of natal dispersal that occurred in that area, how much immigration there was, how old birds were on average, how much turnover there was from year to year. You can see there's lots of small-scale variation in these demographic processes, and what we're relying on is that variation kind of creating the potential for sort of changes in the way demography affects the culture in the population. And we had 3 measures of sort of cultural outcomes in the population. So the first is a measure of cultural diversity, which is just how much variability in the song types there was locally, so from low to high. Another was how unique the local song types were relative to the population as a whole. So an area with low cultural uniqueness would be just songs that are common in the population, whereas high would be songs that are just rare in the population. And the third measure was the degree of cultural turnover, so the change in song composition at a local level from one year to the next. Okay, so this involved analysis of over 100,000 individual songs and sort of rather sophisticated analysis by Nilo, who, if you're interested in this work, I suggest talk to him about it. It was really, I think, a really impressive bit of work by him. But one of the things we saw really clearly is that at a small spatial scale, there is quite a lot of variation in these sort of cultural properties in the population. So, for example, cultural uniqueness varies over distances of just a few hundred meters. The amount of turnover from year to year in the song culture varies in space as well. And we could show that there's also cultural change over time. So, for example, most of these birds we knew exactly when they were born. So if we compared the difference in age between birds, we could show that the further apart birds were born, the more different were the songs that they were singing. So in other words, older birds and younger birds sing different songs that are predicted by the time interval between their dates of birth. So what drives this kind of diversity at the small scale? So one of the things we found is that when you have a lot of dispersal into a neighborhood, you tend to have birds that sing more mainstream songs. And we think this may be because the more that you move around, the more likely it is that you will encounter commoner songs as you move around. So you tend to bring those with you. We looked at what immigrant birds do, so birds that are coming from outside into the population. So rather than introducing new songs, which we thought might happen, they tend to adopt existing songs. But again, they tend to have larger repertoires, which may also be linked to the fact that they're moving further, so they encounter more songs as they disperse. If we looked at neighborhoods that were skewed towards older individuals, we tended to find those had more unique songs, so more cultural diversity, and were more culturally distinct than areas with younger individuals. And if we look at what happens, so what's driving the sort of cultural change from year to year, this is mostly driven by turnover of individuals from year to year. But if we compare conditions where you've got, for example, lots of dispersal and lots of immigration, you'd have much slower cultural change than if you've got older birds in the population. So what this work showed us is that this kind of individual information, combining these individual life history data with learned behavioral variation, we could really show there was quite a strong influence of sort of demographic processes on the way that cultural variation was distributed and changed in the environment. So that's the last of my sort of examples of the application of these studies. So why have these studies been important and successful? Well, I think there are sort of 3 main sort of explanations. The first is that the sort of long-term nature of the work that these studies involve enables you to really think about processes rather than sort of static events or states. You're often working in a system where you continuously build understanding, and that enables you to think about sort of systems rather, the way that processes are interlinked rather than processes as individual entities. Long-term perspective also enables you to analyze change over time and also particularly to appreciate the importance of rare sort of long tail events. So like, for example, the droughts and the Darwin's finches, which was really the most important driver in evolutionary change in that population, but only occurred every 30 or so years. So while they've been successful, where are they going in the future, these kinds of studies? Well, I think there are some obvious areas which I think are going to really massively increase what we can do with these kinds of studies. So, and first of those is, I think everyone would say this, the application of AI in different ways. So actually the song analysis I talked about involved that. We can use AI to classify environments, but I think a really exciting thing is actually the potential of methods for individual recognition using visual deep learning methods. So actually, in a sense, you may even no longer have to catch individuals or handle them in order to actually identify individuals. And that's something which there's already work showing that that's possible in these natural populations. There is the potential for biologging, so fitting devices to birds or fitting equipment out in nature that collects data for us to reveal massively expanded kind of phenotypes that we can study in these kind of populations. So a couple of examples here. One is, this is from repeated tracking of migratory common terns. So the red lines are tracks by an individual in successive years. You can imagine in a decade or so, we'll be able to start to connect that to what its offspring did and so on, and really have a very deep understanding of flexibility in, say, migratory behavior and how it's transmitted between generations and so on. Third future direction, I think, and we're seeing this already, is that people are starting to pool together insight from multiple studies. So there's a couple of examples here, studies that have taken the data from individual long-term studies and said, okay, pool them together and what can we say about the sort of synthesis of effects in populations as a whole. And I think there'll be a lot more of that as time goes on. So I want to just close by talking about some sort of challenges and final thoughts for these studies. So long-term studies of individuals, I think, have been scientifically very, very successful, but they have been conducted on a very limited set of taxa. You'll notice I only talked about birds and mammals, right? And I think that is a major issue for our general understanding of biology, but that actually new methods, so AI, genomics, biologging, could actually change that. So we may be able to do studies of a whole range of different taxa in a decade or two. There are challenges as well, though, right? So one of these is particularly as times are uncertain about science funding. So one is about continuity. So these often require individuals to dedicate a very long period of time, so decades, to one particular study. The other is about funding, of course. You know, the challenges around funding are obvious, and sort of lack of funding could be fatal for a study like this. And then there's a more sort of philosophical issue, which is that these studies are inherently often non-experimental because, in a sense, the value of the study is that it's undisturbed, and experimental manipulation can shift that system away from, you know, the ability to use the data that you've got in the past. So there's always, in a sense, a kind of an issue that people who run these kinds of studies wrangle with, which is how we deal with the kind of, you know, the reviewer request, which is, right, you know, all you've shown is a correlation. How are you going to actually test these things causally? And that's something which I think is still a sort of challenge for these kinds of studies to wrestle with, is when we balance experimental versus non-experimental inference. Okay, so I think it should be obvious to you that these kinds of data, these kinds of studies, they only work if you have fantastic teams of people. This is just some annual shots of our field team in Wytham. We have a particular tree that we climb up, and some years that tree's not been quite big enough to hold all of us, but none of the work that I've mentioned would have been possible without literally hundreds of people's effort and actually, of course, the funding that supported it as well. So thank you very much.
Great, thank you. We now have time for questions. Who wants to get us started? Yeah.
Hello, awesome talk. Thanks very much for being here. So you mentioned this a little bit, that these long-term studies are conducted obviously only on a few populations because it takes so much effort to do so. There's also probably a geographical bias in where these long-term studies are conducted. So I'd love to hear a little bit about your ideas for how to draw sort of these larger scale conclusions from the long-term studies that you've been involved in. You know, I know there are like synthesis papers out there, but and also your thought process on, you know, what sort of caveats do you do you have for this kind of work?
Yeah, well, I think that's a really good point. Absolutely, the point about the sort of geographical bias is very strong. I mean, I think this is a sort of North American, European, and African phenomenon, and the African work often done by people from North America and Europe going to Africa to do it. And there's all sorts of reasons why that might be difficult to kind of set things up in the way that that they were done 50, 60 years ago. I think one of the interesting questions is to ask is, what do we mean by a long-term study, right? Is it simply about time, or actually are there other metrics we could use? So for example, if we think about time in terms of generations, then actually some of the models that have been picked in the past are very poor models, because they're things that take decades to become sexually mature and reproduce, and actually some of the things that might become possible as models, so sort of some large insects or smaller mammals have shorter generation times, and actually if you're interested in evolutionary processes, that might be a different way to get at the same questions, right, over a shorter period of time. So I think we should sort of think about the extent, what do we mean by long term? Is it time? Is it generations? What defines that? So I think that's one way to think about sort of expanding the scope of these kinds of studies. And I suppose what I wanted to try and say is that the application of the combination of people who can do clever things with computer vision, genomics, to actually identify individuals and link them together over generations is something that I think will transform a lot of potential for this kind of work. So you had a third question, I think, or a third part of the question, which was about synthesis. So yeah. Yeah, I guess to acknowledge that we're doing the best that we can with the data we have, which is always the case, right, with any system that we study, and to think about why that might be problematic. I think one of the other things that I'd say is that there's a slight tension at times around ownership of data in this field, and whether data is sort of freely available or not, and that there are some people who, in a sense, are willing to share data very freely, and others who are more. And I think there's a sort of shift happening in this field towards data openness as well, as it is in most fields, and that's a very welcome thing, because I think that will make them more available to a wider set of people as well. So I'm not quite sure I'm answering your question, but I think there's a complex set of factors that sort of determine this. But yeah.
Next question. Oh, Jen, you have one.
Okay, this one comes from someone watching at Zoom. The socially learned information you first examined was quite fitness relevant, knowing how to open puzzle box, while the latter, birdsong features, seemed selectively neutral and perhaps learned during a critical period. First, is that true about birdsong variant in this species? And if so, do you imagine that the conclusions about the importance of population turnover in culture change would be quite different for more payoff relevant cultural traits?
Short answer is yes. Perhaps I should go into that a bit more. So yes, it's true that song learning in Great Tits is not a very short period, but it's learned during their first autumn and winter. It's quite, when you're talking to someone who's online, it's where should you look? Right, I'll look at the main audience. So, and absolutely, the interaction between when information is being learnt and when individuals are moving is a crucial part of this story. And the paper actually talks quite a bit about that, and some of the effects that we see, for example, of immigrants not bringing in unusual songs, but actually just basically learning more songs, probably as they're moving around in the population, can be understood partly as the sort of interaction between when you're learning. So absolutely, the puzzle boxes. So actually, is that fitness relevant? Well, maybe. I mean, actually, the whole aim of that study was to give the birds a puzzle which they'd never encountered naturally, but I guess was something that they would be somehow motivated to learn by having a food reward. So we didn't really set that up to kind of particularly look at sort of foraging skill or success. The idea was that would be a sort of relatively a behavior that actually almost any individual could perform. So actually, as we saw, most individuals did learn that behavior. You may have seen that what happens with that behavior is it doesn't spread to 100% penetrance in the population. You get a sort of between 10 and 20% of individuals that don't display it. And often those are actually individuals that have learnt an alternative behavior, which is that actually there's a detail of the way the experiment was done. We programmed it so that the doors would stay open for a few seconds after a bird had taken a mealworm. And other birds learnt that rather than having to learn to open it themselves, they could follow another bird in. So actually we had this mixed population of, there's a classic sort of model in behavioral ecology, producer scroungers, which developed in this system. So I think in a sense that was an unexpected result actually of the way we set the experiment up. But it was not, I think in principle, any great tip probably could carry out that behavior. So it's probably not that sort of, performing behavior itself is not kind of a sort of selective challenge for them. So,
More?
Thank you. That was a deeply satisfying talk. It was a beautiful talk. But I have a question somewhat tangential probably to your main themes, but I just can't help be biographically curious about it. Do you happen to know how David Lack responded to Richard's critique of Darwin's finches?
no, I don't actually, and I wish I did. I mean, I don't think, I guess Lack was clearly someone who had strong ideas, and he was not afraid to take people on in print, because there was this sort of famous, this very famous criticism of Wynne Edwards, right? He wrote a whole book basically to criticize Wynne Edwards. So, I don't know. I mean, I suppose the point was probably a little bit unarguable actually, because really, to make sense of these patterns, you need to collect field data and kind of, do proper experiments. So I don't know whether he sort of just conceded the point quietly and slunk away with his tail between his legs, but I don't know. That's a really interesting question. I'd love to know the answer to that.
Thank you.
Did you also have a question?
Yeah, I just wanted to ask a question about learning, because I believe true also with chimps. If they learn one way to tackle a situation, that's the way they do it. It's like unlearning is hard. Would you say that's the case?
Yeah, well, so we did look into this a bit in the work that Lucy did, and there are two examples of this. One is that so if you remember, the way we set this up was we had subpopulations where we introduced one behavior and it diffused through. Occasionally most birds stay put more or less in the same place in the woodland. Occasionally birds move a longer distance, and so we think we had in that experiment about 15 or 16 individuals that moved between our replicates. Some of them moved between replicates where the same information had been introduced, but of those that moved between sites where, for example, red was the dominant solution, the majority of them, if they moved to an area where blue was dominant, would then switch their behavior to match the behavior they were seeing. And that actually tied into some other analysis which we did in that paper, which suggests that one of the things we're seeing there, and what explains the very rapid spread of this population and near complete penetrance, is that it's some kind of conformist learning is happening, that learning is positively frequency dependent. So you're more likely to copy the behavior that you see that's commoner in the population, which is at the time a relatively, something which we do, right? How far that extends in the animal kingdom is perhaps not very well accepted yet. So that was, I guess, one thing that showed there was plasticity in the behavior. The other thing we did was we're interested in whether one behavior having become very common, could it be a sort of like a kind of trap that would keep a population away from being able to adopt a new behavior if another behavior became favorable. So once we got populations established in one or other of the solutions to this puzzle, what we did was to shift. So as I said, when we set the experiment up, the payoff for sliding the door either way was the same. So there was no reason to slide it one way or the other. But what we did was to make the payoff for sliding the door to the minority solution in the population much higher than that for the majority. So in other words, there was now a payoff for switching. The question then was, how rapidly would the population be able to shift its behavior back away from this previous majority? And that happened, but not as quickly as we thought it might. So it actually turned out to take quite a long time, so several weeks, and to be a much slower process than the initial diffusion through. So they can shift, and there's a paper, Lucy and Richard McElreath, who I think was here in Davis for a while, and me as a very minor player, which analyses that and tries to understand that in terms of different sort of models of learning. So there is, I'd say the bottom line is there's plasticity and flexibility in that behavior, but it's fairly determined by, for example, stochastic things like which individuals you tend to be encountering. So you have to sort of see a behavior frequently enough in order to sort of reach some threshold to switch your behavior, for example. So processes like that are probably happening. So lots of sort of interactions between what individuals think about the world and what they're seeing other individuals doing that are important.
Ben, I have a question for you. Who are the scroungers? Are they the young ones or the old ones, or males or females?
No, well, when we started looking at this, I was convinced it would turn out to be, kind of teenage kind of mums, it'd be sort of the Daily Mail, which is our kind of, the worst newspaper, would kind of get hold of the story and run with it, and it would be, but actually it turned out we couldn't predict anything. There was nothing that predicted becoming a scrounger in this population. It was actually very striking that once you adopted either scrounging or producing, you more or less stayed in that state. And even if tested a year later, you'd sort of apparently remember, what you were, even though you'd not seen the devices for a year. So we think it might just depend on, sort of some, it's a little bit like your mollies, some, stochastic things that just push you in one direction or another, and then there's a sort of build-up of effects. But we didn't have enough, the data quite of enough quality to be able to sort of, say what it was that had determined that. But we couldn't find anything that predicted which of the states you adopted. Yeah.
If it works, it works.
Yeah.
This is just a follow-up to that question, because I was wondering, and you already answered this, whether if you learned on blue, you could switch to red, and you said that they do quickly, but then you don't switch from scrounging, and that's
interesting. There are some individuals that do switch, but it's much more common that you stay in the same state. Yeah, so it's kind of like once you've adopted that, that's what you're doing. Yeah.
So it's mostly just like a frequency-dependent learning that everybody else is doing red, so even if you were blue, you kind of...
Yeah, so we did want to kind of use, once we realized we had this, Lucy and another postdoc was working in Oxford at the time, wanted to try and use this to see if we were able to sort of switch the payoff from being a producer, the sort of relative payoffs of the two strategies by rewarding one more than the other. Could we shift the frequency of scroungers and produce the population? But actually they weren't very easy to shift from their behaviour. So there's not much individual plasticity once they'd adopted this particular behaviour. So they were less plastic in that than they were with the slide thing. But in a sense, they're quite different behaviours, that perhaps makes sense.
Great. Well, it's 5:15, and so I think that is time. And so if you're joining us for dinner, it'll be right next door. And if not, we'll see you soon. Okay. Thanks, Ben.